Hi everyone, my name's Genevieve. I'm pretty excited about the life science workshop we have on at the Dask Summit. And so I thought it might be nice to tell you a little bit about a project I've been working on recently using Dask. So the data I'm going to share here with you today is provided by some people in one of my old lab groups from Marcus Kitchen's group at Monash University. They collaborate with Stuart Hooper and many others to work on lung development. And so the data we're looking at here is X-ray CT data where there's a microfill contrast agent through the blood vessels that makes them stand out and are really easy to see. So this project, what we really wanted to do was to be able to analyze like the network, the skeleton network that we see with these branching blood vessels in a way that was going to be able to run on something like a laptop. So what we were finding is that even for like fairly small data sets like these, the computational needs of the pipeline, some of the steps in this process were really like memory intensive and it was causing crashes. So we're able to use Dask to kind of keep below our memory ceiling. So I've made an example notebook here that I'll share with you all, turning this analysis that would work for small scale data into something that we could do in a distributed context. The reason skeletons are really interesting is that we find them in lots of places in nature. I'm going to show you some stuff about the blood vessels in a lung today, but these kinds of structures, we see them in lots of places, including animal and plant biology. So like the root structures of plants, the capillaries in leaves, not just the blood vessels, but like say the neuron networks in the brain or the branching of your airways. When I started this project, I thought it was going to be a nice quick one, famous last words, because what I was hoping to do was to be able to use Dask's map overlap function for basically the entire analysis. I figured I could read in the images with Dask image, rechunk them to something that was going to be like a sensible size to let us stay under that memory limit. And then I could do all of my image filtering and thresholding right through the last analysis stage by mapping that with Dask's map overlap. Turned out that it's not quite that simple. So while a lot of the first stages worked really well, once we got to the final like analysis stage where there's a reduction, so we're taking each of the image chunks and computing the branch statistics, the type of result we get back from that is not the same shape as the image chunk that went in. And it turned out that that was causing a bit of a problem. And the second problem that we ran into was that the skeleton analysis library that I was choosing to use, called SCAN, this is written by Juan Nunes Iglesias, while that works really well for like a single skeleton volume, it wasn't written in a way such that the internals were compatible with distributed analysis. Okay, so here is a very simple skeleton. It's just this T shape here. And what we can do is look at this as an array where all of the pixels are 0, and then we can label each of the non-zero parts of the skeleton with an integer ID. We can represent that skeleton shape as a graph adjacency matrix, and that is what this looks like here for that shape. So in a graph adjacency matrix, for each pixel and its neighbors, if they're neighbors, we're going to fill in one of the values in this matrix. The location is going to be governed by the pixel IDs of the parts of the skeleton that we're looking at, and the value that we put into the matrix is going to be how far apart those two neighboring pixels are. So if they're side by side and we have no units, that'll be like a 1. But if the pixels are touching but diagonally, that's going to be like a little bit further apart. So given this kind of like graph adjacency matrix, we can use that to compute skeleton branch statistics. And SCAN as a library is accelerated with number so that we can really get some performance benefits out of this computation. So if we're going to do something similar with Dask, I might show you just with a very simple example that's so small that we wouldn't even need Dask for it. But here I've made a Dask array where each chunk is just a copy of this original like test skeleton. So here we can see them side by side, joined up. It's very, very similar to what we had before. Now we can label all of the non-zero pixels in the skeleton with a unique integer ID. And we can also calculate like the degree of each position in the skeleton. And what that means is just a label based on how many neighbors that point has. That's actually really simple to do with Dask. We can use a convolution. Now, when I wanted to take each image chunk from my thinned skeleton and compute the branch statistics from that, I told you this was where we found that map overlap wasn't going to help us anymore. So it's a bit complex in the way that it concatenates the results back together again, and didn't perform well with like ragged or like uneven shaped outputs, because we don't know how many non-zero pixels are going to be in each chunk of our array. What we get back is going to be like a certain number of dimensions, but like we're not sure about the length, and that was difficult for Dask as it currently is to stitch back together. So to work around this, we were able to use Dask delayed and put that inside a list comprehension to generate the results that we needed. Because of that, we needed to handle our own grabbing of the chunks with some overlap between them. You need a 1 pixel overlap between skeleton chunks so that we can tell whether or not like the skeleton branch ends at that pixel or if it continues just into the next chunk. There's a function in Dask called slices from chunks, and is able to use that as inspiration and modify it slightly so that we could take not just the chunk but like the slice plus some overlap there if there was a neighboring chunk on that other side. So with those two pieces of the puzzle in place, we could iterate through the blocks and pull out the chunks with like that 1 pixel overlap and pass it to our delayed function that's going to actually hand over to scan and do that analysis for us. When we did this, the important thing to remember here that we found is that it's really important to like pass in this meta keyword argument, and that's because if you don't do that, Dask is smart enough to try and figure that out for you, which ordinarily would be really good, except that because it's here in a list comprehension, if you do that, you find that it does this extremely slowly and sequentially, so it's not parallelized at all. And that's really unnecessary in our case because we know the type of result we're going to get back. It's just that we don't know the length of like how many data points will be in it. So that's relatively easy for us to calculate using this Dask data frame util function called makeMeta. And the last thing that we need to do after we've used that list comprehension with the delayed function inside it and concatenated that into a data frame is just to drop the duplicates. Because we have that 1 pixel overlap between image chunks, it means that we're kind of in effect counting those pixels twice. And we need to get rid of that before we put things into the graph adjacency matrix. So the graph adjacency matrix for this tiny little example looks very similar to our non-distributed example. So this is kind of like two of the same skeletons stuck together, which is not very surprising because that's what our original image data was. So when I do this distributed skeleton analysis on like the real data we have with our blood vessels, another problem that we ran into was computing the skeleton branch statistics. This is another area where scan just isn't set up for distributed analysis. There is a function in scan for computing these branch statistics, but it accepts this skeleton object. And that skeleton object class can't be computed, or like you can't initialize it for a really large distributed data set. Our workaround for this was a bit hacky. Really what I was going to try and do here is to just initialize a skeleton object function using some dummy data. So something very small, and then there are a lot of attributes belonging to this already initialized function, and I'm just going to overwrite each of those with the attributes that I've calculated for the real data set. And so if we do that, that actually works quite well. So by using this trick where we initialize the skeleton object in scan using some very small dummy data and then overriding each of the attributes in it with like what it should be from the real data that we've been able to calculate. We've been able to work around some of scan's limitations because it's not quite set up for distributed analysis and get to our final point here with the summary statistics from the blood vessel data in the lung. So you can see that the branch statistics are very different. Some of the biggest differences were really obvious even without graphs. In particular, there were just like many fewer blood vessel branches in the hernia model than in our healthy case. That makes sense because with the hernia model, the lungs are underdeveloped, they're quite squashed, but we also see differences in like the branch distances of the blood vessels that do exist. Yeah, so this was a really interesting project, certainly a very fun one, figuring out the ways we can get around each of those challenges. And it certainly taught us a lot of interesting things, not only about the Dask and the internals of the way that works, but also about ways we could potentially adapt third-party libraries like Scan in the future to support distributed analysis.